Hello, my dear bachas, and welcome back. I know your exams are just around the corner and you all must be studying so hard. But please don't you worry because today we're going to revise the chapter on enzymes in a super simple, super fun way. Come on, let's make sure you nail this chapter and get full marks.
Okay, so let's jump right in. What even are these enzymes we keep hearing about? Well, think of them as biocatalysts. In simple words, they're the tiny, super fast workers inside your body. You know, every single second, lots and lots of chemical reactions are happening inside you just to keep you alive. And guess who's making it all happen at lightning speed? Yep, it's these enzyme superheroes.
All right, it's time to officially meet these superheroes. What are they made of? How do they look? Let's find out.
Now here's the first thing you absolutely must remember for your exam, okay? Almost all enzymes are proteins. Simple. And you know, just like any hero has a special base, like a bat cave, our enzymes have a special place to work their magic. It's this tiny little pocket on the enzyme, and we call it the active site. This is where all the action is.
So how exactly do these heroes get their work done? How do they connect with the other molecules? Well, you can think of it like a secret handshake. And you know what? There are actually two main ideas about how this special handshake works. On one hand, we've got the classic, kind of old-school theory called the lock and key model. And on the other hand, there's the more modern, more flexible idea, the induced fit model. Let's see how our whole understanding of this has changed over time.
The first model, the lock and key model from Emo Fisher, is actually very straightforward. It just says that the enzyme's active site is completely rigid, just like a lock. And the molecule it works on, the substrate, is like the key. So only the one correct key can fit perfectly into that specific lock. It's a perfect pre-made fit. Simple, right?
But then Daniel Koshland came along with an even cooler idea, the induced fit model. He suggested that the active site isn't rigid at all. It's actually flexible. So when the substrate comes near, the enzyme changes its shape just a little bit to fit the substrate perfectly. It's like how your hand fits perfectly into a glove, where you can even imagine the enzyme giving the substrate a nice, tight hug. This is the model we accept today.
Now you know, just like any superhero, our enzymes have certain conditions where they work their absolute best. These are their power-ups. And then there are conditions where they slow down or just stop working altogether. They're weaknesses. So let's figure out what makes these enzymes happy and productive.
First up, we have temperature. You'll often see this shown as a bell-shaped curve. At really low temperatures, the enzyme is kind of lazy and slow. As you raise the temperature, it gets faster and faster and faster until it hits its peak performance. We call this the optimum temperature. For most enzymes in our bodies, that sweet spot is around 37 degrees Celsius. But be careful. If you make it too hot, the enzyme literally breaks apart and loses its shape. We call that denaturation, and then it's game over for the enzyme.
Next is pH. Enzymes are very picky about this too. Again, it's a similar bell-shaped pattern. Most of our body's enzymes love a neutral pH, somewhere between 6 and 8. But there are always exceptions. Take pepsin, for example. It's the enzyme in your stomach, and it absolutely loves a super acidic environment around a pH of 2. So you see, every enzyme has its own favorite pH level to work in.
And finally, there's substrate concentration. Just imagine you keep giving your enzymes more and more work to do, more substrate. The more you add, the faster the reaction will go. But, and this is a big but, it only works up to a certain point. Eventually, all your enzyme superheroes are busy. Every single active site is occupied. They're working at their absolute maximum speed, which we call Vmax. At this stage, even if you add more substrate, the speed just won't increase. The enzymes are totally saturated. Okay, Bacha Party, pens down, eyes on the screen. This is a very common exam question. I can almost guarantee you'll see this. It'll say, define Km and explain its significance. Let's break this down right now so you never ever forget it.
So what on earth is this Km? Well, the Michaelis constant, or Km, is simply the amount of substrate you need to get the reaction going at half of its maximum speed, so at Vmax divided by 2. Now, for the significance part, a low Km value means the enzyme has a very strong attraction, a high affinity for its substrate. It means our superhero has a really strong grip on its target, so it doesn't need a lot of it around to get to work.
But what happens when a villain shows up on the scene? In the world of enzymes, these villains are called inhibitors. They are basically substances that try to slow down or completely stop our enzyme heroes from doing their very important job. Let's see what their tactics are.
So, the first type of attack is something called competitive inhibition. In this case, the villain, the inhibitor, looks a lot like the real substrate. Because they look so similar, it actually competes for the exact same active site on the enzyme. You can imagine it like you and your friend both running to grab the last empty seat on the metro. Only one of you can get it. The inhibitor blocks the spot and the real substrate just can't bind.
Now, there are two main types of these villains. We've just met the competitive inhibitor, the one that fights for the active site. But there's also a non-competitive inhibitor. This one is a bit more sneaky. It binds to a totally different spot on the enzyme, and by doing that, it changes the enzyme shape so it can't work properly anymore. This table you see here, it's your cheat sheet for the exam. For competitive inhibition, Vmax stays the same, but Km goes up. For non-competitive, Vmax goes down, but Km stays the same. Make a note of this table, Beta. It's practically a guaranteed question.
Let's just focus on that competitive villain one more time. Why doesn't Vmax change? Well, it's because you can actually fight back. If you just increase the amount of the real substrate, you can outcompete the inhibitor. You basically flood the area with so many of the good guys that the villain has a much lower chance of getting to the active site. But because you now need more substrate to reach that halfway speed, the Km value goes up. It makes sense, right?
So we've seen our heroes in action, we know their powers, and we've even met their enemies. But all this working and fighting, it takes a lot of energy. So where does all this energy come from? Well, that brings us to the cell's ultimate power source.
All that energy comes from a very special molecule called ATP, which stands for adenosine triphosphate. This is such an important molecule, it's known as the universal energy currency of the cell. A good way to think about it is like this. Whenever your cell needs to do any work, it has to pay for it, and the money it uses is ATP. It's like the cell's own UPI or Paytm wallet.
So how does the cell spend this money? Well, an ATP molecule has 3 phosphate groups all linked together, and the bonds holding them are packed with energy. So whenever the cell needs power, it just breaks that last high-energy phosphate bond. Pop! And just like that, ATP becomes ADP, adenosine diphosphate, and a whole burst of energy is released for the cell to use for whatever it needs.
And how much energy are we talking about here? It's a very specific number. When 1 mole of ATP breaks down into ADP, it releases exactly 7.3 kilocalories of energy. That's a really powerful little packet of energy, always ready to be used anytime, anywhere inside the cell. All right, let's do a super quick rapid-fire revision before we finish up. Are you ready? Okay. Enzymes are our protein superheroes. The place where the magic happens is the active site. The modern theory for how they work is the induced fit model. They are very picky about temperature and pH and have their own optimum levels. Inhibitors are the villains trying to stop them. And all this hard work is paid for by the cell's energy money, ATP. You got all that? Fantastic. And I'll leave you with one final thought, just to make you a little curious. We've talked about enzymes being the workers, right? But what happens when an enzyme isn't just a worker, but it's also the manager? The master controller that regulates an entire chain of reactions? Well, that's a story for another time. You all have been absolutely amazing today. Study well, stay confident, and I know you are going to do wonderfully in your exams. All the very best.